Sebastian's originally from Germany. He got his bachelor's, master's, and PhD degrees from the University of Bremen. And then he decided to come to the US for the first time. Did a postdoc with Al Sporman's group at Stanford University from 04 to 08. And then he went back to Germany and assumed what would be termed an assistant professor position at the University of Tübingen, where he was from 08 to 2014. Now he came back to the United States, and I hope he's here to stay. He's been with us ever since. And he is an assistant professor, or associate professor in our department right now, non-tenured. But we hope he will soon be a tenured associate professor. But that's part of the point of this presentation. And I would say that no pressure. But if I had to describe, and of course you can look on his website and see how he describes himself, but Sebastian is what we call a microbial ecologist and a molecular biologist. He uses cutting-edge molecular biology tools to study environmental problems and really investigate the mechanisms of how things work, and basically how bacteria can be exploited to clean up, for example, mining waste or agricultural runoff. Or wastewater for that matter. So he's really doing very nice fundamental work to try to really dig at the mechanisms so that when you try to engineer things better, you really have the fundamental tools to do so. And just one example, he was involved recently in a paper that came out actually 1 year ago today in Nature Communications on the topic of biochars. And these are basically pyrolyzed biomass that has been used as soil additives. And he's been doing some really fundamental work with colleagues in Germany on how biochars work for, say, nutrient retention, soil fertility, things like this. This paper came out, again, just 1 year ago today. It's already been cited 18 times in Web of Science, and it's in the 97th percentile in terms of altimetric score. So now, of course, there are 260-some thousand papers published, so it was at 7,000th overall. But still, good. So anyway, I'll leave it to Sebastian to take over. Okay.
Thank you very much, and good morning, everyone. I'm very excited to be able to present here this morning. After this phenomenal introduction, I would have to point you to another presentation I'm giving on October 23. So if you want to hear about biochar problem and the paper that apparently gets very well referenced right now, I point you to come to an agricultural technology symposium of the Midwife group, which is on the St. Paul campus, October 28th. So today I have prepared something else to give you an overview about the topics I'm working on and also the main questions I'm mostly interested in. And for that reason, since we are a very interdisciplinary department, I start out with a little broader to give you a little bit more of a perspective of what gets me so excited about my research, and then delve into some projects in a little bit more detail to tell you how we approach questions of addressing how microbes actually interact and work together. So I put this provocative title out, and while preparing my presentation, also the main reason I'm doing my work is I try to understand communities so I can help operators that operate engineering systems to manage these communities successfully. The point of this presentation is to tell you that I think we are not quite there yet. So I would like to change it a little bit to say studying microbial communities, and I tell you exactly what is missing in order to actually successfully manage these communities. Okay, I will start out with a broad introduction. What is molecular microbial ecology? I tell you a little bit about the challenges and approaches to study microbes. I will talk about two very recent projects that are just submitted and published this year of two of my grad students. And then I finish up with a vision about what I will do in the next 5 to 10 years with respect to advancing, especially the approaches to studying microbes and to get to the point that we can actually successfully manage them. Okay, microbial ecology. If you leave this seminar and are not convinced that microbes are everywhere and determining all life on this planet, I didn't do a good job. So if that is the take-home message you get, that's my goal. We are living on a microbial planet. Everything on this planet is run by microbes. You will probably appreciate that they feed the world. Right? If you like cheese, bread, anything fermented foods, that's all based on microbial activities. You will probably also appreciate that they had a huge impact on human history, not only because they're causing diseases in humans, but then also plant diseases that affect people in different countries. For example, the Kennedy family came over to the US from Ireland because there was a misharvest of potatoes that got infected by a fungi, so that it basically starved 1 million people over there and caused a huge immigration to the United States. However, these are mainly examples of what you might experience when you are interacting with microbes through disease that are negative. However, if you think microbes are bad guys, then think again. At the end of this presentation, I hope you will appreciate how valuable these organisms are. They are the engines, the best chemists that we have on our planet, and biochemical engines that drive this planet. They make the oxygen we breathe. The most abundant cyanobacteria, the most abundant photosynthetic organisms are cyanobacteria in the ocean. They provide a lot of the oxygen we breathe. They recycle the dead. If you take microbes out of this planet, we would just be piling up organic waste and it would not be degraded. They fix nitrogen in association with plants. They help animals digest food. And we use them to produce renewable fuels, bioproducts, and pharmaceuticals. And I'm interested in how we can use microbes to control pollution. And I'll give you a couple of examples how I approach microbial communities involved in these processes with different types of techniques. And more so, and that is most exciting right now, also in the field of microbial ecology, it shifts over. The same techniques we are using, we can use to study human microbiomes. And right now, the perception is, yes, there are incidences where we get infected and get disease and sick through the interactions of microbes, but most of the time, they keep us healthy. So to give you one example here, our body has as many microbial cells as human cells. So you're only 50% human based on the cell numbers, right? And this gets shifted. So every time you go to the restroom, this kind of like ratio shifts a little bit. If you look at how many genes you have in your human genome, it's about 25,000. All the genes you found in your gut microbiome are 3.3 million. So the metabolic diversity that you're carrying around that is not actually human is tremendous. And it will change radically how we think about treating disease or health states in the future. Just one last example on this. There are 30 bacterial metabolites in your brain. And there's a connection between your gut and your brain. So there is room that if you have a wrong diet, let's say, it could cause depressive symptoms. And there will be way more of that, how microbes actually affect us as human beings in the future that will revolutionize medicine. Now, this is a medical field. I'm very excited to be able to use the same tools that I'm using for studying organisms that degrade pollutants in the environment that can also be used to study humans. That's why we have this close relation here. The point here is, all these examples that I've given, already Pasteur said, if we would take out microbes from this planet, life would be possible. And I refer you to a very nice article in PLOS Biology by Jack Gilbert and Neufeld, who said, what would happen if you could wave a magic wand and take all microbes from this planet? And what the consequences would be. So life without microbes is, in the end, the conclusion is, after a year, life would come to a halt. We would get severely affected by digesting our food. we would need to nutritional supplements, and of course we would suffocate in all our waste that is not be degraded anymore. Animals that depend on bacteria that fix nitrogen would die, and so on and so forth. Okay, why haven't we yet studied this tremendous biomass on our planet with this detail that it deserves? Because most of the time we can't see it. However, they are small in size, these organisms, but large in numbers. You see, the estimates go 10 to the 30 microbial cells on Earth. It involves atmosphere, ocean, Earth's crust. It makes up to 50% of all biomass. So all higher plants and animals are the other half. These organisms are around for 3.5 billion years on this time scale. That's when the first microbial cell is supposed to be evolved. They have been taking part in the development of oxygen and oxygenic atmosphere, so they turned our planet from a reduced into an oxidized system. And here you see only about 2 million years, depending on how you count it, there's humans around here on this planet. So that means the time these organisms had to evolve just created this huge diversity, phylogenetically and metabolically. And that is what we can explore for biotechnological application, for environmental applications. And that fascinates me. There's not a thermodynamically possible reaction, I would claim, or that is also kinetically possible, that is not exploited for energy by microbes on this planet. They can breathe solid metals, like iron oxide. They can breathe chlorinated solvents and reduce them in this process. Something that's actually toxic for us, or something that is solid, where you would think of, how do I get this dissolved? How do the organisms actually reduce this compound, so placing electrons on that? No matter, microbial biology is a very recent study. It's a very novel object of studies. And I'm rushing through the history here very quickly. In 1687, Antony van Leeuwenhoek improved the microscope and found for the first time, oh, there's single tiny little organisms everywhere. I look. And then there came people like Pasteur and Koch, Bayering, and all others to develop the decoding of the genetic information. What is important for my field is, in the 70s, DNA sequencing was invented, and then in 1985, Carrie Mullis got the Nobel Prize for the polymerase chain reaction that allows us to amplify genetic information. And that was the birth of molecular microbial ecology. In 1986, this is the first paper that defines the field by Norman Pace, Stahl, Lane, and Olsen. They published, we can use the PCR technology to amplify specific phylogenetic marker genes that are in all life and use that to find out what microbes we have in environmental samples, how many we have, and study the composition of these communities with molecular techniques, because we cannot grow most of these organisms in the lab successfully. We are trying this for 150 years with classical microbiology techniques. Only 1% of all organisms we have detected by genetic techniques are actually in culture. If you go to the American type culture collection, France, Japanese, there's about 12,000 strains in there that you can order, like from a catalog. But there's supposed to be 50,000 or 500,000 organisms out there in the environment that we know of because we find their genes, but no one is able to grow them yet. So we have to study these organisms' techniques that let us allow to look into these organisms, not only who is there and how many, but then also what these organisms are doing. Microbial ecology currently rewrites biological textbooks. If you go to the library here and pick up the biology textbook, look at the first page on evolution, you will find a phylogenetic tree in there that is wrong. This one is from 2016, and I have another one from 2018 that is based on genomes that have been sequenced for organisms, from all organisms on this planet. The eukaryotes down here, this is us, higher plants, and all animals. You see the diversity of this group fairly insignificant, actually, compared to the archaea and the vast diversity of bacteria. We have 2/3 of bacterial diversity, 1/3 of archaeal diversity, and the latest tree, if you include another branch here, which are called Asgard, Archaeota, then the eukaryotes become the sublineage of the archaea. So we're going back from 3 domains of life to only 2. And this is just what happened last year. And every year there's new metabolisms identified that revolutionize how we think about microbes and their capabilities, that let us rewrite, let's say, the nitrogen cycle in our textbooks. And the reason is, if you look at the branches here, where you see the red dots, these are the lineages where we don't have any cultural representatives. The majority of this tree, to us, is unknown. We don't know how they look, what they do. Never had anyone had these organisms in culture, could study them in the lab. So we need to look at these organisms in more detail with techniques that allow us to study them without going there in culture. All that, phylum over here, candidate phyla radiation, these are nanobacteria, endo and episymbionts that sit on other bacteria. No one knows how these organisms interact. And that's my point here. We need to look not only at who is out there. We can make these trees over and over and over again. We can count organisms over and over again, but we need to study how they interact. Microbes in the microbial communities are the ultimate social network. They talk to each other, they interact with each other, they influence their behavior based on who is around them and with whom they partner up or with whom they compete in the environment. And that's what we need to embrace, and only then we can study microbes or manage microbes successfully. That's my point here. Okay, so microbial ecology is the scientific studies of interactions, basic principles that determine the distribution and abundance of organisms. And as I've shown you already, since its first time that people saw microbes through a microscope, the discipline has always been driven by technological developments. DNA sequencing, PCR, nanoscience, microscope improvements helped always to advance the technology. The field is driven by new techniques. Now, developing new techniques in my lab, and this is part of my vision, I would like to tell you something very cool that we recently developed that will help us understand more about the interactions of part of these communities. So the first 2 questions driving this field are who is out there and how many of what kind. This is a very exploratory study. Some people don't always think, oh, why don't microbial ecologists come up with more hypotheses? Well, in the beginning, we didn't know at all who is out there. So most of it was, well, there's a cool sample, like a hot spring. Let's find out who is there and how many of what kind to fill up the tree, these databases of sequences. So we have this information now. Now we can go beyond that and look at what these populations are doing, how they're interacting, how these population size functions are regulated, and then we have the tools at hand to manage them successfully for, let's say, certain functions that provide a service to society, like successful wastewater treatment or improved wastewater treatment, or treatment of agricultural runoff, mine water treatment, etc. Okay, so this is an image of a wastewater treatment plant. We are supplying this project. This technology is successful for over 100 years. But we're using very conventional parameters to operate these plants. pH, temperature, oxygen reaction potential, retention time, and it works. However, we know what organisms are in wastewater. They have been sequenced. They are all in this tree. We know how many of what kind. But then sometimes processes fail. And then ask an operator if he could explain what happened, or how he could have prevented it, foreseen it, regulated it, managed it. And there's no clue. So by just filling up databases with sequences, we will not get that information. We will have to study how communities are assembled, how they get to a state and how they maintain it, how resilient they are, how they change over time. Microbes evolve tremendously fast. It's a moving target. It's not something like you will be able to study today and it will be the same in 10 years from now. These communities are changing. And we're not recognizing that we need to look at the spatial scales that these microbes live in, at the microenvironment, how they actually associate themselves in, let's see, these aggregated sludge flocks, and how these species interact with each other. And that's part of what we are trying to do in a project in collaboration with Tim, that is the PhD thesis of the graduate student. We are going in, looking at a sequencing batch reactor, at a wastewater treatment plant in Brainerd, and study the phenomenon that is quite common here in temperate climate zones, that is nitrification failure in the winter, and what is going to happen. This is the central slide. I will come back to that 3 times to make the point that we need integrated approaches. Our genetic tools, metagenomics, DNA sequencing, are tremendously valuable. They provide a lot of information, but they don't tell us much about who's interacting with whom. I can sequence your genome, but I won't be able to read out of this genome who your Facebook contacts are. There is interactions that are important to understand what you're doing in your behavior, how you react, but it's not in that information. I need other information for that. And that comes not from the top-down approach, so basically sample a whole community, sequence everything, find out who's there and what they are doing, how many we have, but we need to understand what they are doing. And therefore we need single-cell resolution, we need imaging techniques, we need to see what is going on, we need to see how they are structured and form these aggregates and how these actually interact with these cells. So I will be giving you 3 examples. The first one is the nitrification failure in wastewater treatment. That is about spatial and temporal resolution. The next one is where we integrated a top and down and bottom up approach using these techniques and genomics to study a microbial consortium. And then I give you a short overview about the technical developments we are doing right now to improve these method integration, to learn even more about the microbes and how they interact together in high resolution real time and on the activity side rather than the community composition side. So the first project, as I said, is a project from my graduate student, Julie Johnson. This is, we went out and studied the wastewater treatment plant in Brainerd. You see here, since the plant was renovated and restarted, the temperature profile and the ammonia effluent profiles plotted in this graph. As you see that as soon as temperatures drop, effluent ammonia concentrations spike up. The plant doesn't do anything wrong, it's not regulated here in Minnesota in the winter, because no one knows actually what's going on and how this problem could be prevented. Other very conservative parameters that are constantly being measured, biological oxygen demand, phosphate concentrations, pH, and others, suspended solids, they don't change, but these are the main 2 parameters that change over time. So we wanted to find out what is going on with these 2 communities involved in nitrification during the seasons. And we created the most comprehensive data set yet collected for one single wastewater treatment plant. I'm tremendously happy that we had good colleagues over there at the Brainerd Utilities who were able to basically give us samples of activated sludge every week for over 2.5 years now. I'm showing the data set for over a year, but we have way more in our freezers. So they go out, sample 3 parallel sequencing batch reactors every week that freeze the samples, and then we look at these communities to create a tremendously valuable data set that has a spatial and temporal resolution to look at exactly how these communities change over time, how they assemble themselves over the year, how they drift and change with time. So this is the Brainerd plant. It has 4 reactors, 3 are always operated at the same time. So we have parallel replicates internally. We have 55 weeks over a year of samples right now. We did 16S RNA amplicon sequencing, created over 40 million reads of organisms, and then classified these in 1,984 unique species level operational taxonomic units. That means these are, you can think of them as species if you want to, but microbiologists don't have a good species concept. So we like to call them just, categorize them just based on their similarity of certain gene sequences. If you look at the overview here, what changed over the year, it actually looks like all of these colors are different types of organisms. It looks very similar. But then if you study that in more detail and delve into this data set, you see very interesting processes underlying the community change with temperature that is related to the loss of nitrification. What we saw is there is a core set of organisms that is abundant year-round. 114 of these organisms. They are always there. They change in abundance, but they are always there. And these organisms make up 75 to 85% of the complete community. Then there's organisms that are only there in a couple of seasons, only in the summer, winter, spring, fall, and they change, but they don't make up so much of the complete community. And then there's a vast diversity of organisms that just pop in and out. They just come in with somebody, so they say, hey, I'm here around for a week, and then there you go. So that is the most of the diversity. So we need to dissect that in order to see which ones are actually important for us with respect to the functional processes that they have. Now, if we look at this a little bit differently, these are all the 3 reactors. There was 1 reactor of failure, so it got started up. So this is why we have 4, but 1 and 4 are actually 1 continuous reactor that just had a failure during 1 week. So these are the 3 reactors over the year, and we basically reduce the diversity of these samples now to 1 point in order to be able to compare it better. Now, if we color from which months these samples come, then you see here the summer months, spring months, they are in the bright colors, and the winter and autumn and fall months, they are in the blue colors. So we see that these communities seem to be different in the summer than in the winter. And if we put on our ammonia data now to see, okay, when did ammonia concentrations actually go up in the effluent water, now indicated here by the larger dots, then you see that this goes up in the winter. There's a strong correlation. If we plot this graph now with the first principal component against temperature, so you see this clear trend of the summer communities, then the fall changes, the temperatures drop, ammonia concentrations go up, these communities are down here, and then they develop back up to a summer state. So there seems to be a drift and a community change in this. And if we just compare the summer 15 to the summer 16 samples that are up here, you see, first of all, this trend, but then you see also the summers are different. And that's the value of having 2 more years of these samples. We will be seeing how they react over the years, how they change through 7 years when we get to sequence these samples. Interestingly, if we look at very high resolution at only the nitrifying communities, these are organisms involved in ammonia oxidation. They are fairly low in abundance, and they don't change that much over the whole year. We try to correlate this with statistical tools with the ammonia temperature variations, and there's no significant indication that there's a correlation between these organisms and the process failure. So when these organisms stay around, the failure can't have anything to do with their abundance changes that had been suggested previously. Now, there are tons of organisms that correlate very well with the effluent ammonia concentration, temperature changes, and we have now a set of candidates that positively and negatively correlate with the temperature changes. And one of these most interesting species here that changes between 2 and 10% almost is the Sulfursporidiaceae, but no one has any indication yet if they are actually involved in nitrification or not, or if they provide a service to the community that might affect nitrifiers that don't change in abundance in their activity. This was on DNA level. Now, Julie had a great idea, and she seemed to be liking to hang out in wastewater treatment plants. And so she took a sleeping bag and slept next to a sequencing batch reactor in the fall, in the winter, in the spring, and the summer, and sampled every 10 minutes, like crazy, in order to get a higher resolution of what is actually going on, and then look at RNA, which is the express information of genetic information. It tells us more about the activity. And here, these organisms, again, seem to have different responses to the reactor cycles. We have the static fill, the mixed fill, the react cycle. We have the settling phase, where we look at the top and the bottom, so the bottom sludge and the liquid on top of that. And again, also the nitrifying organisms here, the dashed lines down here, seem to be having no differences in the activities in the different seasons. However, if we look at a general marker here, the CNS, as expressed sequence, which gives us a value for the protein synthesis activity, that drops drastically in the winter. So organisms change their behavior in the winter. It seems to not be the nitrifiers. And we can identify these organisms now, because Julie is just getting ready to submit these sequences for sequencing. So we will know who that is. And they are not the usual suspects. They seem to be something else. And since we can compare that to the previous data set I showed you, where we already did the correlations of species to temperature and ammonia concentration, we will be seeing if these are the organisms that we already have on our list to check right now. So we did this, we looked at core and transient species, we saw how much of these populations are changing during the seasons, we correlated this to the plant activity, and we will have soon the information to look in genetic tools at the structural information that we have for the specific organisms that are actually changing their activities over time. We collected samples to do microscopy. Alongside, we have the freezer full of that, and we will be able to identify these processes more specifically on a spatial level by using genetic stainings of these organisms in situ. So we saw in ammonia oxidizing bacteria maintain population abundance and actual activity, but however, there seems to be the general protein synthesis activity of bacteria seems to change in the winters, and we need to know what these species are. And there's a strong correlation of some of these OTUs with temperature and nitrification performance, and we need to find out what this role is in nitrification, how they're spatially organized, and then we will know how they actually interact with this. So we are on a good way. Julie has 1 year left of studies. This first part has been submitted now. We're working on the RNA part, and the last one will be completed in the next year. And I think we have a tremendously better insights then into what is actually going on in Brainerd in this specific plant during the winter, and why this activity is lost, and potentially then find out what we could do to manage these communities in the fish. Okay, switching over to another topic here, in which we not only use sequencing, but then also imaging and cultivation on the single cell level. I will therefore tell you a little bit more about the microbial iron cycle. Microbial iron cycle is driven by organisms that can freeze iron-3 minerals and reduce it to ferrous iron. And then there's organisms that can use oxygen under microfluidic conditions and light energy to oxidize iron too. But then there's organisms that are fascinating. These organisms use nitrate to reduce iron 2. So they have an electron donor, which is iron 2. They reduce nitrate in their process. And these are autotrophs. So this is a very interesting technology. It's an autotrophic denitrification that creates iron minerals on the side, like down here. Some cells encrust themselves with iron minerals. This is this part of the iron cycle. There's many biogeochemical reactions in the iron cycle, and the biotic reactions. We're looking here at a sediment, for example, where we have iron 2 and then nitrate available, but no oxygen, and in that zone, these organisms are supposed to be active. I was lucky to get hold of one of these cultures that can do these reactions. Actually, I'm very attached to that because it was extracted from a little freshwater pond in my hometown in Germany in 1996. And since then, this culture is being cultivated just with iron 2 and nitrate, no organics. And it maintains its community structure. We looked at who's in there. We saw some dominant species are these iron oxidizers here, or organisms that are related to them, and a denitrifier, and then some other species at lower abundances that seem to be cooperating in this culture to maintain this process. What we wanted to know is how these organisms work together to perform this process. And that's how we approached this by sequencing the metagenome of this culture, and then also by looking at isolates and used imaging mass spectrometry to study these organisms and their activities in more details. So we have a very good autotrophic nitrogen pollution control technology here. It's all over the literature right now, very recently. It could be used for groundwater remediation. People think because you're forming iron oxides in the presence of nitrate, then you can also complex arsenic species and remove them from groundwater together with these organisms in potential biofilter settings. John Coates looked at this as a technique to enhance microbial oil recovery because these organisms make iron minerals in tight pore spaces and change the hydraulics in these underground systems that could enhance oil recovery. And it has also been recently suggested, just in March this year, as a potential mechanism that could actually explain why there could be life on Mars based on the interactions of ions and iron oxides and iron 2 and nitrate. So it's a tremendously interesting process, but we don't know exactly the mechanisms. Is it one organism? Is it two? Is there a biotic reaction? Is there an abiotic reaction of the nitrites with the iron 2? Who is doing exactly what? So we tried to isolate organisms out of this culture, and we've been successful to get 4 of the partners, but the one that was the most important to us seemed to be not growing. We tried everything that we could, microaerophilic conditions. However, once we have the genetic sequences, we can trace it and stain it just because we know some of its genes. We don't need to grow it in the absence of the others to study what its capabilities are. Now, then we grow this culture with everyone in there. It oxidizes the iron, it reduces the nitrate, cell numbers increase. Now the isolates, they don't oxidize iron, and they don't reduce the nitrate if they are alone. So it seems to be only associated with this one member in this culture. You see here, this is the culture in the beginning, this is at the end. You see all the iron oxides formed. Here's the nice rusty colors. All the other cultures seem to be not able to do this. There's always a little bit loss of iron 2 because you get this precipitation here on the sides, and since there's phosphate in the media, you get a little bit of hydrogen formation, which can be oxidized by these organisms, but they take a little bit of the iron 2 out. But these organisms are not autotrophs, these other members of the cultures, and they don't oxidize the iron. However, if you get them an organic substrate like acetate, they can reduce nitrate. Now, what we did is we teamed up with Eric Rowland in Wisconsin and studied 2 of these cultures and sequenced the metagenome. We got some genomes completed, others of the less abundant species not completely assembled. And then we used an imaging mass spectrometry technique on the single cell level to use isotope labeled substrates, 13C bicarbonate, 15N ammonia, to study the uptake of these compounds into the species and then how they change and exchange these compounds among each other. So this is the results from this part. I will just summarize it on this one slide and showing you what the experimental evidence is we got from cultivation and numbers and what we predicted from the genome data. So this species is capable of fixing CO2. It's a true autotroph. It makes organic compounds. Bradioisobium, based on its genome, it can fix CO2, but it doesn't in culture. We didn't find any of the activity of uptake of certain C-labeled bicarbonate. Interestingly, the autotroph can grow on acetate, which is actually not predicted from the genome, but through metabolic studies. The heterotroph can can take up acetate and oxidize them. If some of the carbon that is fixed by the autotroph is used by the heterotroph, we were not able to show. We think it has something to do with EPS metabolisms, but that would require further studies. The Gallionella species, predicted from the genome, can reduce nitrate to only nitric oxide. Nitric oxide is super toxic. Now we come to the point: why are the other species around? Because they're complete denitrifiers. They can take nitrate to denitrogen gas and basically detoxify the nitric oxide that the autotroph produces. On the other hand, the Bradioisobium feeds ammonia to the Gallionella species because it's also capable of disseminatory nitrate reduction to ammonia. And we showed the uptake and the genes in the system of this, in the genomes of the Bradioisobium. This is our true iron oxidizer. This organism doesn't do iron oxidation, but they depend on each other for the complete metabolism. This organism would kill itself by this end product, and this organism wouldn't be able to survive without this organism fixing carbon and supplying that carbon for the Bradioisobium. So tremendously important interactions that you cannot only derive when you look at genomes. You need to study substrate uptake, you need to cultivate organisms and look at what is going on with activities on a single cell level. Now this brings me now to my last point. How can we improve this? This study I just showed you took 4 years to perform in order to cultivate, isolate, sequence, analyze the sequences through the nanosems measurements. And the number of cells we can analyze on a single cell level with nanosems is very, very low. How can we improve this? How can we make interaction studies of microbes more high throughput and better accessible? So we need real-time functional screening and monitoring. This single cell is the ultimate unit in which we have everything combined. Genomic information, activities, and we get the chance to look at them under the microscope. However, the heterogeneity among these cells with respect to their activities is tremendous. This is a plot from our isotope labeling studies. Each of these dots is a single cell with respect to its uptake of 13 C-labeled carbon and 15 N-labeled ammonia. You see a huge difference of organisms in their different growth phases, exponential phase, stationary phase. Some of these cells encrust, some of these cells don't encrust. So we need to appreciate this heterogeneity. On the other hand, we need to be able to study the population activity, considering all this heterogeneity that makes up these populations. For that, we need to increase our throughput. We need to miniaturize our reactions, and we need to run reactions in parallel. Right now, this is what we are all doing in the lab. We have these microbial plates or small little tubes, and we run our reactions of organisms in that. This is how sequencing is done. This is how DNA amplification, PCR, is done. Volumes are 1 to 10 microliters. If you're lucky, you get your PhD student to run 1,000 samples, maybe 10,000 samples, but you really need to shake and force them to do this many reactions. But it's not enough. We need more. And therefore, we need to do it smaller and smaller and smaller. And that's what we are trying to do here. We are integrating droplet microfluidics and fluorescence-activated cell sorting in order to make reaction volumes that are only 2 to 50 picoliter, which is 10 to the minus 12 liters. We can run reactions of up to over 1 million, and then we have access for single cells with enough statistics. And the important thing is, if I want to study the genomes, I need less contamination. There's so much free DNA around. If I run this in these high volumes, then I have a lot of background information that actually doesn't belong to my species. If I reduce the volume drastically, or a couple orders of magnitude, I can decrease that contamination. So what we are doing right now is we have that micro droplet formation set up here. So high-speed camera, several pumps. Here we have a tiny little ship. This is an image of that ship. The junction size here is 30 micrometers, and we make these tiny little reaction vessels. And here you see how fast they are produced. We make about 4,700 of these per second of a droplet volume of 50 picoliters, and the diameters of that reactors is 13 microns. If we use our reagents for genomic amplification, PCR, or growth medium, we can incorporate this in these droplets together with cells. And that's what we're doing. We encapsulate single cells in these droplets. We run DNA amplification reactions to identify subsets of organisms based on their phylogenetic and functional marker genes. We then encapsulate them the second time to make a double emulsion, to have this whole system again in an aqueous solution, and then we can run this through a FACS, through a sense-activated cell sorter, in order to study which organisms we have in these reactions and which one performed, let's say, have a certain metabolism going on, or we're active with respect to substrate convergence. And this is how this looks like. This is our first approach to optimize the system. This is how homogeneous we can make our droplets. This is a mixture of two species, E. coli and Shibonella. E. coli has a glucuronidase gene here that Shibonella doesn't have. This is 16S gene all organisms have. So every droplet that had a cell lights up in green after DNA amplification through a PCR. Only the E. coli cells are the red ones, and then you have an overlay here, and it's probably a little bit hard to see, but there are these ones that have the red and the green color. They are yellow. And based on these fluorescent signals, I can sort this droplet and assess subsets of populations. But this is just one genetic quick screen to look for organisms that we can cultivate that might have a certain function, let's say ammonia oxidation in wastewater. However, I can use this system also to incubate cells with substrates, with antimicrobials, or to put two species together and see how they grow. And then I can sort them, I can screen for them, I can quantify them, and in the end, I have them in these tiny little containers that I can also use to run DNA amplification reactions and actually get access to the genomes. So I get integration, high-throughput activity assays together with genomic information on a single cell level, and that with access to statistics, and because I see every single droplet and their activity on the flow sorter, I have access to the heterogeneity within the population. These are just a couple of applications that we are working on right now with people at the Biotechnology Institute to develop these technologies further for screening of huge genetic libraries, directed evolution experiments, and then what I'm mostly interested in is these activity-based physiological screenings, where we will be able to use biogeochemical substrates in these containers and see how organisms convert that, and then sort these cells based on the activity, and then amplify the genomes later on to find out who that is, and link this information of activity and structural information. With that, I would like to thank, first of all, my group. The wastewater project was run by Julie. The organisms that can oxidize iron and denitrify was run by a student that I started out working together with in Germany, that came over here and worked for over half a year with me here at the University of Minnesota. The method development is done by my postdoc Che, and of course all these other students support us, support me, and greatly in the science. I'd like to thank Tim and the public utilities at Brainerd for their support on the wastewater project, Paige and Mihai for being my mentors in the department, and of course everyone else here in the department at BTI. It's a tremendous, great experience to be able to work here. And what I've been able to pull off in the last 3 years was just also because of your tremendous support and interactions and collaborations in the last years. Funding came from these funding sources, And you see us here standing in front of a picture which was taken at an escape room. So I would like you in your labs, and I would like to challenge you. If you take your lab to this escape room, and you get out of this room faster than we, I admit you did a better job, but we got out of this room 2 minutes time to spare, 1,300 points. So if you want to be better, take your people over there and let us know if you can rescue the world from a viral infection before it's too late. I like that a lot. In the end, I'd like to thank you for your attention, and I'm happy to answer your questions. Thank you.
Hi, Sebastian. Thank you. I enjoyed that very much. I guess I have a specific question that I almost want you to give a general answer to. You had trouble isolating the Gallionella.
Yeah.
And your data suggests that you have the production of a toxic nitric acid. Did I get that right?
Yeah.
Do you think that's sort of a general problem with culturing organisms, that they produce a toxic metabolite that it's hard to, I guess, experimentally remove as you go? So could you just comment on the culture?
Yes, definitely. I mean, as I hopefully got across with this presentation, is that there's interactions on certain levels that we don't completely understand yet in these communities. And part of that could be that organisms compete with each other, produce toxic metabolites that inhibit the growth. So in order to study these organisms more effectively, not the ones that would kill themselves, but maybe ones that are affected by others, we need to get them in isolation, and we can increase our culturability maybe by just looking at single cells in these tiny little reactors. So yes, I absolutely agree that this could have been a problem of getting these organisms to grow. Usually you would expect they grow in gradient tubes with oxygen. If they are closely related to other Gallionella or zero-oxidant species, but this organism doesn't seem to do that. It needed the nitrate. And so it needs a partner around to take that away. It's a classical contract of that you also would find in syntrophy. Certain organisms, secondary fermenters, would not be able to get energy for their reactions because it's an endergonic reaction that they ferment ethanol. If not, a methanogen, for example, would constantly take away the hydrogen to keep the hydrogen partial pressure low to make this reaction energetically favorable. Here we have another example of that. It's not energy metabolism. It's energy metabolism for this one organism, but it results in a toxic compound that needs to be detoxified by another species. That decreases our chance of cultivating many of these organisms, including that one.
I've got a question about the one organism that you pointed out that you found interesting in your wastewater system.
Yes.
Could you use your single cell sorter to? Is there a way of determining chemically using some sort of labeled substrate whether that organism can in fact be nitrify?
Yes. Yes.
Other than just the genetic, I can understand how you can do single cell sequencing and figure it out that way, but can you actually label and how would you do that?
Well, the problem with sequencing is you find only what you know, right? So if we would if we get the genome of that organism, and we can through our technologies, and we don't find ammonia monooxygenase in this genome or anything that is homolog to that gene, we will not have an indication that this organism can actually oxidize ammonia. So we will need to run physiological assays. And one of these experiments being performed to look at Kumamox organisms used a specific fluorescent dye against this homolog of the ammonia monooxygenase that can stain activity of the enzyme.
Okay.
So we think we could combine that with that screening assays we have to look at the genome of these organisms and see if it actually be able to get it in culture. However, it might also be not a direct involvement in ammonia oxidation that affects the whole community. These organisms are known to be filamentous organisms, and they support the sludge flocculation and aggregates. So many of these interactions depend on that organisms are tightly spatially associated, and we will have the samples available in freezers right now to look at how the sludge flocks look in the summer, in the spring, and in the winter. And maybe their structural integrity without these aggregates falls apart. And it might not be a direct involvement in nitrification, but part of that disrupts, let's say, the interactions between ammonia oxidizers and nitrate oxidizers. And that has a community effect on the overall activity. So that's what we are missing if we only look at genomes, but that's what we are trying to get.
Excellent, thanks.
Have a question?
At the wastewater treatment plant, I'm wondering to what extent seasonal variations in dissolved oxygen with temperature have on which microbial metabolisms are favored?
Of course, there's a lot of that impact. They operate granite at a very constant parameters. They don't change the sludge retention times and their oxygenation profiles during the seasons, and then they also don't heat up the air. Which made this plant perfectly for other studies. But there is these conventional parameters that you can use to modulate a little bit of the nitrifying activity, which has been done in other systems.
Hey, so I was wondering if you think it might be possible to take that Gallionella that produces the toxic NO and put in the genes to make the enzymes to, get rid of that and convert it, and so that it doesn't need a microbial partner, and that in general, when you're finding these microbes that only live in communities, is there some beyond the simplicity reason why you wouldn't want to just take all those enzymes and engineer one super microbe that can do all those reactions in one place?
Yes, and that's part of the bottom-up approach that some of my colleagues follow up on, the genetic engineering of strains, to create new species by putting assembling genomes that can then be active for a specific function. However, for that, right now, the classical genetic tools require that you can grow these organisms. Most conveniently, not only in liquid, but on an agar plate to pick individual colonies. And these organisms, specifically the Gallionella species, they only grow in gradient tubes, if at all, and our species didn't grow in all conditions we tested so far. So that would limit this approach for now. But yes, I agree, it is an attempt in many of these colleagues that I have worked on engineering organisms to perform certain functions better. However, my personal view is that a mixed community management is always more applicable and successful than trying to get a single organism in a reactor, which I need to keep sterile, put energy in to prevent other organisms from contaminating these systems. So I'm believing in the mixed community approach where I know enough about the interactions of these species to manipulate them in my for the intentional functions instead of putting new organisms together and raise them in isolation or place them back in the environment where they would not be able to compete with the rest of the community.
Anybody else? All right, let's go to that.